This is video 1103, section 11.3 in your textbook. This is other patterns of inheritance. Last term we studied Mendel's principles, and we learned a couple of rules that occur with the rule of simple dominance, or the principle of simple dominance. Well, there's exceptions to these rules, and then there's exceptions to the exceptions. So what exactly happens if an allele is not completely dominant over another? Or what if there are several alleles for a gene? So we're going to look at those in this section. So one exception to Mendel's principle is that some alleles are not dominant, nor are they recessive. Another one is that many genes exist in several different forms, and so they're said to have multiple alleles. So it's more than just a big T and a little t. There might even be a C. And then lastly, sometimes traits are produced by the interaction of several genes. And we're going to look at all three of those in this section. So firstly, these are three main exceptions to Mendel's principles, and one of those is that many genes have more than two alleles. Many genes have more than two alleles. So for instance, blood types. You have an A allele, you have a B allele, and then you have an O allele. Many traits are controlled by more than one gene. So sometimes, like for instance, hair color, there's so many different phenotypes there that there's different shades of brown hair, different shades of blonde hair, and that's because they are controlled by more than one gene. So sometimes if one is present, it might block the other one. And so, oh, I'm sorry, not hair. It's going to be fur, dog fur. Those are usually controlled by more than one gene. That's why you can have two dogs that have brown hair, and they can have puppies that have white hair, that have like a yellow lab. They could be a brown lab. And then lastly, using Mendel's principles alone cannot predict traits that are controlled by multiple alleles or multiple genes. Type of complex inheritance I want to talk about is incomplete dominance and codominance. They are very similar, but they have distinct differences. So with incomplete dominance, this is when you have neither allele is dominant. Neither one is dominant. And so what happens is when you have the heterozygous condition, you have a third, not a third phenotype, but the phenotype is a blending many times of the two main phenotypes. So here we have the F1 generation, which we have a true breeding red one and a true breeding white one. Okay, so here's our true breeding red one and our true breeding white one, and they produce the heterozygous condition is pink. Okay, so in this case you have a red crossed with a white, and then all the offspring are pink. Alright. So this heterozygous phenotype is an intermediate, so you have a third phenotype. So as far as in problems, you might see it written where big R, they're all capital letters since neither one is dominant nor recessive. And some books use what's called the prime symbol. Okay, so you might have, since you have the red ones, the white ones are written with this little symbol here, and that's the prime symbol. So anytime you see that, you know that we're talking about incomplete dominance, and that's where you have the third phenotype that is a mixture of the two. So in order to get pink flowers, you have to have each of the original phenotypes. Now in codominance is very similar, but in this case, instead of them blending together, you see both of them. So both of the alleles are clearly expressed. So an example is this chicken here. So he has white feathers and black feathers. He doesn't have gray feathers. He has black and white, because the allele for black feathers is codominant with the allele for white feathers. The name for that is erminette, or the checkered chicken. So it'll be speckled with black and white. Sometimes you might see a white flower with little red flecks in it. That would be another example of So both alleles are expressed in the heterozygous condition. Example is this checkered chicken. So a black chicken crossed with a white chicken is going to give you a checkered chicken. Okay. Now if you notice, we used the same letters here in the codominance, didn't we? Whereas with the incomplete dominance, we use the same type of letters, but it makes it a little bit easier when people use that prime symbol with the incomplete dominance. The next type will be multiple alleles and polygenic inheritance. So a gene with more than 2 alleles is said to have multiple alleles. The main example that you need to know about is human blood type. There's 4 types of blood. You have type A, type B, type AB, and type O. Alright, so this is what determines what your blood type is. Remember the carbohydrates on the cells? Well, they had these little cell markers. Remember, they were used for cell communication or cell identification. You also have proteins on the cell. Well, antigens, these antigens here, antigens are proteins. And so a person that has type A blood would have the A antigen on it. B blood would have the B antigen. And then A and B would have the A and B antigens. And then O would not have any antigens. Now, the I stands for a term called immunoglobulin. Immunoglobulin. That's what the I stands for here. And we use the I because the big I and little I are dominant and recessive. The big I is dominant, the little I is recessive. So here's our little key here to show you how this works. So for a person to have type O blood, they have to be homozygous recessive for that I. For a person to have type A blood, they have to have immunoglobulin A, or immunoglobulin B would be for type B blood. So since A and B are codominant meaning they're both expressed, right? Codominant, okay? And then I, or little I, O, is recessive to both of these, okay? So the symbol for A-type blood, is going to be IA. Symbol for B will be the IB, and then the symbol for O will be little I. Okay? There should be an O right there. Example of multiple alleles would be coat color in rabbits. So look at so these are all different alleles. There's an allele for light gray, dark gray, chinchilla, that's where it's kind of gray and white, and then Himalayan, which is the black and white. In this case, we have what's called a hierarchy, where the gray is dominant over all of these, okay, and then the C is what we use for our dominant allele. So, excuse me. So C with, there, sorry about that. So chinchilla is recessive to gray. Himalayan is recessive to chinchilla and gray. And then albino is the recessive to all of those. So for instance, if a rabbit was a big CH, what color would it be? That's right, it would be gray. If say you had a big C and a little c, it would still be gray, because what? Because gray is dominant to all of these. Okay, so it's 10 possible genotypes and 5 different phenotypes. Or 5 or more phenotypes, I guess it says. So the genotypes could be C, CH, CC, CC, CH, CCH, C, little c. Okay? An example. The presence of multiple alleles increases the possible number of genotypes and phenotypes, which is what I just talked about. And so this big C, the gray, is going to be dominant to chinchilla, Himalayan, and albino. Now, with the polygenic traits, at height. Say these were 3 sets. We were looking at height. So this person would be a medium height because they have 3 dominant ones and 3 recessive ones. So who would be taller? A person with 3 dominant ones, 2 dominant ones, or 1 dominant allele? I'm sorry, dominant genes, yeah. Yeah, the person with 3 dominant genes. Now if they had 6 dominant genes, they'd be even taller. Where here? Who would be the tallest on here? The tallest would be this one here because you have 6 dominant genes there. Okay, and then the shortest would be this one here because they have 6 short genes. And then everybody else is in between here. And so the way you look at this is you count to see how many of the dominant genes they have, and that would be determine what end of the spectrum for height that they would be on. So, in general, most people are going to be here, right? That's the medium one. This would be the tallest. This would be the shortest. So, if we were looking at, say, skin color, which is what this one is with, you got 6 of the recessive ones here. So that'd be the lightest color. Then here you have 6 of the dark alleles, or the dark genes, and that would be the darkest color. Because if you look at this, 2, 4, 6, 8, 10, 12, right? So that, but the shortest it can be is 4. So 4 plus 12 gives you the 16. And I would give you this. Don't think that you'd have to figure that out. But hopefully you were able to figure that out. Here, each of the uppercases alleles is worth 2 centimeters each. So, in this case, this is what our offspring were. So we have 4 centimeters. And how many uppercase alleles do we have? We have 1, 2, 3. And each is worth how much? 2 centimeters, so 6. And that gives us the plant being 10 centimeters tall. How that works. So does the environment have a role in how genes determine traits? Yes, it does. Gene expression can be influenced by the environment. So they can affect or not. So sunlight and water can influence the phenotype. So think of phenotype of a plant. If there's sunlight, the plant will grow and it turns green and all those type of things. Temperature can affect it. If it's too hot, the plant can shrivel up. If it's too cold, the plant can shrivel up. A good example here is a Siamese cat. Their fur turns darker in cooler conditions. Now, if you remember on the exam, one of the experiments I gave you was the fur color in rabbits. And they put ice on the rabbit's back to see if the fur would grow in dark. Same thing happens with these Siamese cats. That's why their nose and their ears are dark, and their paws, and the tip of their tail. Could be, think about your weight. You might be predisposed to be a certain weight. But obviously if you overeat, that kind of overrides what your genes are, and it will change. Here's an example of the western white butterfly. The ones that hatch in the summer have different color patterns than those in the spring. Okay? So the ones that hatch in the spring have more pigment. They're a little bit darker than the ones that hatch in the summer. In this case, the environment influences which butterflies express their wing colors. Or, excuse me, what color is expressed. So in order to fly effectively, the body temperature has to be between 28 and 40 degrees Celsius. More pigmentation allows the butterfly to reach the warm body temperature faster. And in the summer, since it's hot, they don't need that extra pigmentation to get warmer. So that would be the adaptation or the advantage that they would have.